The correct answer is D. The appropriate components to prepare the primary working reagent, that means the carbol-fuchsine, for acid-fast staining are basic fuchsine, phenol, methanoethanol, and distilled water. The correct answer is C. The appropriate quality checking method of a newly prepared and received acid-fast stain is by performing a quality control checks using the appropriate control samples containing known positive and negative samples. In this case, these could be slides with known Mycobacterium species that should react with the Zen stain. The correct answer is A. The most appropriate reagent that can effectively replace the gram stain's counterstain called safranin when stauched out is by using neutral red reagent. Note that neutral red is selected as the counterstain because it stains well Gonococcus and Meningococcus. The correct answer is C. The most appropriate technique that should be followed by the laboratory technologist to prepare the smear for gram staining from the collected purulent body fluid specimen is by centrifuging and using the sediment part of the sample. Because the sediment contains the concentrated cells, including bacteria, which are necessary for accurate identification of causative agents. The correct answer is B. The most appropriate steps should be applied on the smear for resulting. The white-yellow glowing rods are by applying auramine phenol stain on the sputum smear. Because the auramine phenol stain binds to the mycolic acids in the cell wall of the acid-fast bacteria, and when exposed to ultraviolet light, these bacteria will fluoresce white-yellow against a dark background. The correct answer is D. The information that was missed from the reported test result was the bacterial arrangement, because the arrangement of such type of bacteria is in a Diplococcus form. So the correct reporting form of this gram stain result will be many gram-negative intracellular Diplococcusine HPF. The correct response of the student should be option C. It stains bacteria. The primary stain, crystal violet, used in gram staining procedure, is a basic dye, cationic, whereas the cell walls of both gram-negative and gram-positive bacteria have a net negative charge under most physiological conditions, i.e. anionic. Therefore, these opposite charge interacts and imparts a purple color for all bacteria initially. The correct answer is C. In this case, the most appropriate preserving method of the urine specimen for later processing will be refrigeration from 4 to 8 degrees Celsius. Also, it is possible to preserve the urine specimen by using boric acid for later processing if delayance not avoided. But refrigerating at 4 to 8 degrees Celsius is good for preserving the urine specimen as compared to using boric acid, because it is simple and does not require any additives. The correct answer is C. In this question, the assessed aspect is sterility. Because the prepared culture media was selected randomly and incubated without any inoculation of organisms and checked for the presence or absence of growth of organisms on the media. These media checking procedure is called sterility checking. Thank you for watching. Subscribe and like our videos to get more videos related to medical laboratory science.